Good evening, everybody. Welcome to BrisScience for July 2023. Thank you so much for coming out on this lovely wintry night, as wintry as it gets in Brisbane anyway. My name is Joel Gilmore and I'm your MC this evening, and welcome to BrisScience, the series of free public lectures on science brought to you by the University of Queensland, where we aim to bring not just the best scientists, but also the best communicators to share their research with the scientists and the passionate general public of Brisbane, and of course everybody watching on YouTube. Hello. So tonight we're going to get into it in a moment, but I would like to start by acknowledging the traditional owners of the lands in which we're meeting tonight here at the Edge, State Library of Queensland, and pay my respects to elders past and present, and acknowledge the ongoing contribution of the first people of Australia to our knowledge, and particularly tonight we're talking about medical science, their understanding of the land and all of those unexplored flora and perhaps fauna of Australia that might still hold important contributions for medical science. So tonight, if we're going to take... Let me try that again. We're going to be... I saw the QR code and I thought I would seize it while I could. So tonight we have a presentation followed by questions, Q&A, which we're going to do over the question app that we have through UQ. So you'll see a blank screen up there. It's close. So you're going to be able to scan that QR code or that Windows desktop. Now there's a chance to look at what they're looking at on their web browsers. Oh, they've managed to get it there. There you go. So you can scan that QR code into your phone, and a great opportunity to turn your phone to silent at the same time. And then after the presentation, I'll come up and we will get to as many of those questions as possible. But never fear, there will be the opportunity afterwards to join our speaker outside, and we're going to put on some food and drink, so we would love for you to hang around and enjoy a proper cultural experience. Should be just as fun as going to the footy with just slightly fewer people. Okay. So our speaker tonight, is my great pleasure to introduce Professor Ernst Wolvetang, who is from the University of Queensland, and he is the Senior Group Leader at the Australian Institute for Bioengineering and Nanotechnology. And amongst many projects, he and his group work with stem cells. And stem cells are those incredible cells that are a little bit like a first-year uni student, full of potential with their whole future ahead of them, and sickeningly optimistic. And Ernst uses every trick in the book to use these cells to understand the big and hard questions, like how diseases affect our brains, particularly as we age, as well as a couple of interesting side projects, which you will see. So to fill our brains with knowledge of brains, please put your hands together and welcome Professor Ernst Wolvetang. Okay.
Let me see, does this work? Yes. Good to see you all out here. Fantastic. I thought I was the only one that liked stem cells, but it doesn't seem to be the case. So that's great. So what I want to do tonight is first of all educate you a little bit more than what you just heard about what stem cells are. And I'm going to tell you a little bit about how we use brain organoids as a model to understand disease and aging. And then I'm going to do tell you where this field is going in some weird and wonderful ways, and then I'm going to end up with a dessert of mammoth for you, okay? Let's see how that goes. So the first thing we should probably start with is a definition of what is a stem cell. And unlike a first-year student, a stem cell divides and produces an identical daughter cell to make two of them. But it also has an option to turn into a different type of cell type in a process called differentiation. And that basically is the definition of a stem cell. And you know stem cells, you just don't know it that you know it. If you go in the morning and you see these skin cells coming off your skin, you don't think about where the new ones are coming from, but they come from stem cells in your skin that make new ones. As you're sitting here, you're making 2 million blood cells per second in your bone marrow. That's stem cells doing that work for you. So you don't realize, because it happens at time scales that we don't recognize. But stem cells are really fundamental to keeping our bodies looking pretty much the same. And having said that, and I'm looking at my audience, as we're getting older, and me too, things are starting to look different over time. And that's one reason is that stem cells are failing. But another process that I'm going to talk about is senescence, and that is also related to stem cells. So let's dive into that, right? So first of all, not all stem cells are created equal. If you think about a mature cell, a skin cell that doesn't turn into anything else, and the cells that I just talked to you about, as the cells that continuously replenish our bodies, they are called adult stem cells or mature body stem cells. And we have those, and they are made when we were born and as an embryo. But then there's a very special stem cell called an embryonic or a pluripotent stem cell. And that one is really the Swiss Army knife and jack of all trades, because it can make every single cell of the body, and it will divide indefinitely, which is kind of handy for research. And they make those more committed stem cells. Now, we always thought that development was going in one direction, from an embryo to an adult, until this guy, Shinya Yamanaka, in 2012 got the Nobel Prize for showing that you can turn this clock back, you can reverse that clock, and you can take a mature cell back to its embryonic state. And so what, you say, like, that's a nice party trick. Why is that such a nice thing to do? Well, for that, you need to first understand development, and I can't get enough of this movie. Because this shows you when you were a 5-day-old embryo, just after you implanted in the uterus of your mom. And as you can see, this disc in the middle, that's going to be you, believe it or not. And they came from these pluripotent stem cells, this small clump of cells in what's called the blastocyst. And then you can see here this line, and I'm going to ask questions about this later, is forming a brain on one side and a tail on the other side, will become your spinal cord. And at that stage, it starts to show 3 different cell types called mesoderm, endoderm, and ectoderm, which basically generates your muscles and your heart, your brain and your skin, or your glands and your gut and your lungs. And then this beautiful process happens, right? I mean, then you just make an embryo and you have a human being. I mean, to me, that's still one of the most amazing things that nature has devised. And it all starts with a clump of stem cells called pluripotent stem cells. And that's why I like them, because they can clearly make a whole human being, all the cell types of the human body, because otherwise you wouldn't be sitting here. So Waddington thought about it as follows. He sees life as a ski slope. You start at the stop in this blastocyst, and then cells make decisions as they run down the green slope or the blue slope or the black run, and then they become different cell types. So what you can do, what's called reprogramming, is take that mature cell types and bring it back up to a day 5 embryo, and now push it into the ski run that you want it to go. So I can start with a skin cell or a blood cell or a cell shed in your urine or a hair follicle cell, as long as it divides. Be careful what you leave behind tonight. And I can reprogram that back to a cell type that was the same as you were, a day 5-old embryo, and then I can make every single cell type of the human body from that cell type. Now that is a cool party trick. Now one thing you can do is you can start to make brain cells, because for medical science, most people can freely donate a blood sample to look what's wrong with your blood, or sometimes even a liver biopsy. But I haven't met anybody yet that says, like, yeah, just take a piece of my brain, see what's wrong. It doesn't happen. So that's why making brain cells from pluripotent stem cells is a really good way to go. Now, as you can see here, these brain cells are growing on a plastic dish in 2 dimensions. And I know, and you can probably see, they are confused. They are moving about, trying to find the right spot, and they go like, no. And the reason for that is because they're growing in 2D. That's not how a brain develops. So we took the red pill many years ago already. Believe me, some of the new generation have no idea what I'm talking about. But if you take a stem cell that you have educated to become a brain-committed stem cell, and you allow it to interact with itself into a small clump of cells. These are these stem cells. And you allow them to do their own thing, then they will actually start to create all the different cell types and all the layers of the brain in an orderly fashion at the time that happens during embryo development. It blows my mind. That is pre-programmed in these brain stem cells to do this all by themselves, and they will make a three-dimensional structure that actually looks and behaves, as I'll show you, exactly, in this case, as the outer bit of your brain that you do your thinking with at the moment, the cortex. That's really what sets us apart from all the other less smart primates, I guess. Maybe the dolphins have a bigger cortex, other story. But here you can see that these brain organoids, and this is a young one, has a stem cell compartment and it starts to make all these different cell types in the dish. That's a pretty good thing to do, because that allows us to interrogate where things go off the rails when there's a disease. So my entire lab is dedicated to do this particular sequence of events. We harvest a blood sample, we make these what's called induced pluripotent stem cells, these artificially made stem cells that were similar to when you were a day 5 embryo. We then make them either into 2D neuronal cell types, but we now just go make brain organoids at scale. And that allows us to do a number of things. One, because I can do this from any cell type from you, I can first interrogate you or ask you the question, what kind of disease do you have? And you go like, well, I've got Parkinson's disease. Then I say like, okay, then you must have genes that predispose you to getting Parkinson's disease. But where does it start and how do we investigate it? I can take a cell from you, a blood cell, bring it back to day 5 and make a brain organoid, and lo and behold, it will start to show signs of Parkinson's disease. And I can apply that trick to almost any neurodevelopmental or neurodegenerative disease that we wish to study. And that is your DNA, that is your genetic makeup. It's not a mouse, it's not anybody else's DNA, it's you. So that allows you to do personalized medicine. So treatments, identifying treatments that only work for you or similar people that have similar DNA makeup to you. And that's important, and I'll tell you why. But not right now. This is just an overview of the kind of programs that we run. We are interested in how these defective genes cause disease. I'm not going to go into the details, but we're also interested in how environmental factors, such as viruses or a lack of oxygen during the giving birth, lead to developmental abnormalities. That causes severe problems down the track. And there are also diseases that are both environmental and genetic. And that's the beauty of making brains in a dish. You can actually impose those external factors really easily on top of genetics that you know and that you can manipulate. So that's a really powerful combination. Now, why do we do that? Not just because it's intellectually and academically stimulating, but also because it makes economic and healthcare sense. Because classically, this is how drug discovery is done at the moment, is you start to screen thousands of compounds, first on cells, and then in mice, and then in primates, and eventually you will start to do a phase 1, phase 2, and phase 3 clinical trial. Many years, billions of dollars, and what do we get at the end? Drugs that are only partially effective in about half of the people. That's just not good enough. So why don't we just skip this mouse bit, and why don't we immediately go to organoids that are of specific individuals, screen the drugs on those models, and then validate them in animal models. Saves a lot of animal lives, it's much more specific, and it's cheaper and faster. So that's why we do what we do. So one thing that we can, for example, do is to test how you can deliver the correct copy of a gene that is defective in a kid that has a neurological disease. And this is an example where we delivered a virus to deliver a gene that is a problem in this kid, And we can test this out, whether it's safe, how much do we need to deliver, does it go to the right cell types, and is it effective? So that's where we're at. Now let me give you a couple of examples of how we use this brain organoid technology to really start to make a difference in people's lives. And I want to tell you about this project called the Persist project. And that stands for Personalized Epilepsy Genes with Stem Cells. Anyway, let me explain it to you. At the moment, when you are diagnosed with epilepsy, and there will be somebody in the audience that has it or will know somebody that has it, because 1 million Australians will develop epilepsy over their lifetime in Australia. It's a lot, it's common. You go to a neurologist, and he or she goes like, well, let's try this. And then it doesn't work, let's try this on top. And then try another drug on top. And then remove the first one and add a fourth one. And you can go for years finding a medication that actually works for you. And the evidence shows that the longer you try, the more likely you are to be resistant to the drug, even one that may have been effective at the start. So this kind of hit and miss and wet finger approach, even though our neurologists are absolutely first class and world class in Australia, that's not good enough. So what we have done is we have made these stem cells from 60 patients here from Queensland that are resistant to drugs and have epilepsy. And then we make brain organoids, and then we interrogate them with mini EEGs, as I will show you. Then we screen drugs on those organoids, and we're just about to test those drugs in real life, and these people will have a diary and see if it actually is more effective than random therapy. So that's personalized therapy, right? That's personalized medicine, and I really like that. And to let you know, we do this in a blinded fashion, so we don't know what they're actually resistant to or sensitive to. Let's see who wins. So, if you think that we are doing this, you know, in the hoods with people just making organoids, no, then we don't do that anymore. We use robots to do that, right? They do it much more consistently. They do it at scale. We can image these organoids over time, we can do quality control, and we can make hundreds and hundreds of organoids of an individual. And that's what we need if we want to start testing drug combinations. Do the math, right? 1 times 2 times 3 times 4 combinations, that's a lot. So you need a lot of organoids. And we can do that using robots. And I should acknowledge the University of Queensland here for investing in the Australian organoid facility that now starts to make these organoids, not just for us, but for other stem cell researchers at UQ, and also from other universities here in Queensland first, and then nationally. Democratizing brain organoid research, and I think that's really important. Okay. So, I'm going to also give you a little bit of science today, okay? So these are markers, and I'm not going to go in what they are, but this, you can see day 25, day 50, day 100, and another day 100 organoid. We section it, we make a slice out of it, we stain it with antibodies to specific proteins that are expressed in a specific cell type only, and then we ask how many are there, are they in the right layers, are they next to each other or not? And I'm not going to go into this, but let's say here you can start to see glutamatergic and GABAergic neurons. It doesn't tell you anything, but these are neurons that are activating and that are inhibiting, because our brains are networks of neurons that continuously activate and try to inhibit each other. And you need that if you want to study epilepsy, because that's where epilepsy sits, a loss of inhibition or an overactivation of activating neurons. Okay, so then we do the equivalent of an EEG in our organoids. So you probably know an EEG. You go to your neurologist, you get the electrodes on the outside, and they measure your brainwave activity. And you can see all these lines go up and down. There are different frequencies, and from that you can see if you have epilepsy or not, especially if you have an epilepsy episode. So we do the opposite. We put our brain organoids and we put them on the electrodes. And we've got 4,000 electrodes sitting on the bottom of this little dish, and then we can start to read out this electrical activity. Because I told you that these brain organoids are doing the same thing as your brain. They are thinking by themselves, okay? And we can also give them seizures. So here is an example of that. So here's patient 70. As I said, we don't know what drug they're responsive to, but that's how they look like normally. So these are electrodes, and this is time. And you can see these neurons are firing, whole conglomerates of them are firing at the same time, just like what's happening in your brain right now. If you give a proconvulsant, so something that will give you a seizure or that will give seizures to mice, you can see that the amount of firing goes up. It starts to fire more quickly. And then we add this well-known anti-epileptic drug, levetiracetam, and that is gone. So that's an effective drug response. However, when I take a different patient doing exactly the same thing, baseline for aminopyridine, give it a seizure, add levetiracetam at the same concentration, nothing happens. So to me, that's our prediction that this patient will be resistant to levetiracetam. Now we're testing all the different combinations of drugs, but you get the picture here. That's how we do drug screening in vitro on the patients themselves. Now then we're also interested in why they are drug resistant or what is actually going on. So here what we do is single-cell RNA-seq. Now let me explain what that is. So remember that each of these dots is a cell in our brain organoids, and we want to know what genes they express. So these genes tell you what kind of different neurons they are, and also whether that neuron is a little bit different from what it's supposed to be. So I compare it to looking at a chocolate bar and saying like, okay, that's pretty good chocolate, and it's probably orange flavor, to looking at M&Ms and looking at each individual M&M and say, okay, that's a red one, that's a green one, that's a blue one. And then we count how many there are and whether all these M&Ms are the same. So that's how we do it. So what I just told you is that we can start to look at genomic variants. So we can capture your DNA. We can look at connectivity and patterning of your brain in the dish. We can look at drug responses, and we can start to look how cells change when they see a drug or they are unresponsive to a drug. Okay, so can we do that with the brain? Or can we also start to look at other parts of the central nervous system? And remember, I was going to ask you questions. Do you remember that disc where the brain was formed from one side and the spinal cord from the other? Where you saw that the spinal cord was starting independently from where the brain was generated. So what we're now doing here is we're patterning those stem cells in the stem cells that make the spine from the tail side, and we allow them to develop. And why do we do that? Because we're interested in children that have a defect in what's called myelination. So this is a disease called hypomyelination with brainstem and spinal cord involvement. Basically, these kids end up in a wheelchair by 5 or 6, usually don't survive beyond late teenage years because their motor neurons, they can't move, they continuously degenerate. And the reason for that is that because there's an insulation layer around your nerves called myelin, just like an electrical cable that insulates it from the rest, and that allows really fast electrical transmission. These kids have a problem with making the insulation around their nerves, hypomyelination. And that's why they get leg spasticity and cognitive impairment. So what we did is we made spinal cord organoids. And we started to look at where this insulating layer is, and these are cells that are making that insulating layer in orange. And to our surprise, again, if we make a brain organoid, that insulating layer is on the outside, just like in your brain. But when we make spinal cord organoids, that insulating layer is on the inside, just like what you expect. So they pattern correctly. They are chock-a-block with cells that make myelin. They have motor neurons in them, here marked by a protein called CHAT. And so we're kind of convinced that these are really pieces of spinal cord, human spinal cord, that we've grown in the dish. Now, this is the first time that anybody sees this, not published yet. So what happens in this disease then? Well, these are control ones where you can see that these cells are on the outside where they're supposed to be. Here's a patient where these cells are in the middle, where they're not supposed to be, and here's a patient that doesn't make them at all. And this is the most severely affected patient. So we can see that the cells that are making this insulating layer are not in the right spot, and the neurons, the motor neurons that make that transmit the movement in your spinal cord, they are properly patterned here. Clearly something is not going right here, right? They're all bunched up together. Okay, so you can pick a white matter disease in vitro, but does it, again, an organoid also has to recapitulate the function of that particular organ. So here we put it again on a multi-electrode array. Here's a control that normally fires, and this is really blew our mind. Now what we saw was boop, 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 nothing, boop, 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 nothing, boop, nothing. Just like spasticity, remember? This is what spastic people exhibit. They can move, and then all of a sudden they lose control. My God, our spinal cord organoids are doing exactly the same. So we're now screening drugs to see how we can inhibit that, and we're also looking at gene therapy to cure this disease. Just keeping an eye on the time. Okay, so we call them axial oligodendrocyte spinal cord organoids. Scientists love these kind of long names. But the important is that they replicate this white matter polarity and they model spasticity in the dish. I bet you didn't think that you could look at spasticity in the dish. Okay, let's change direction a little bit. This is for the elderly under us, like me. So I'm really particularly interested in studying brain aging. And not just because I'm getting old, but because most of the diseases that are the scourge of our society, like Parkinson's and Alzheimer's disease, the biggest risk factor for that is age. Not your genes, it's age. So can we study that with brain organoids in a dish? Well, yes, I'm going to show you can. But just to remind you what happens in nature, when a leaf gets old, it undergoes a process called senescence. And senescence is a natural process that happens in nature and in our bodies, and is designed to prevent cancer. As soon as a cell starts to misbehave, our body goes like, okay, either get rid of it and kill it, or just stop it from dividing and prevent cancer. And then it becomes senescent. Or if its DNA is too much damaged, it becomes senescent. The problem with it is that as soon as a cell has become senescent, it starts to tell its neighboring cells to also become senescent. And that's why they're called zombie cells. They are like the zombie apocalypse. They have 4, 8, 16, 32, it just goes exponential. So that's why senescent cells are a problem, especially during aging. So let me first tell you a first story about this particular protein, Clothol. Clothol is the goddess that spins the wheel of life in Greek mythology. And Clothol is also the name of a protein that if you make too much of it, and you live in Iceland in a very special family, you don't get Alzheimer's disease, and you're guaranteed to live to 110. That's a kind of a cool protein to have if you make too much of it. And indeed, when you engineer mice to overexpress this protein, Clothol, they live 30 to 40% longer than their litter mates. Interesting. That's an interesting protein. The problem is that it's a really complicated protein. It's anchored in a membrane, it gets processed, it's expressed in different forms in different tissues, and nobody's been able to figure out how it works in the brain. Aha, I hear you say. Ernst is going to look at brain organoids. Yes, I am. So that's exactly what we did. We make brain organoids, and in this green color, this is a stain for senescence. I'm not going to go into the details. There's more of this stain as they become older and older. This is a marker for cells that no longer divide, P21. There is more of that over time. P16 is a bona fide senescence marker that goes up after week 10 or 13, exactly the time when Clothol decides to drop its expression, to really go down. And we're going like, okay, that's not coincidence. That's got to have something to do with each other. Now, to test that, we do a trick. Again, this is like a biology trick. So we take an enzyme, Cas9, that that's driving the CRISPR revolution. This is an enzyme that we hacked from bacteria, that has a barcode. We can direct it anywhere in our DNA that we want. But in this case, this Cas9 has been inactivated. It doesn't cut anything anymore. But we still have the barcode global positioning system of this protein. So we can direct it, and we can fuse it to a protein that activates a gene. We can direct it to the promoter, the engine that drives Clothol expression, and when we add it, we can see that in the presence of the inducer, Clothol expression goes up when it normally would go down. And then what happens with senescence? That's basically totally gone. No more senescence. So Clothol inhibits senescence in the human brain organoid, and when you drive it. We published that. So my analogy is like, so if zombies are the problem, then this is preventing the zombie apocalypse, right? This is making sure that they don't become senescent in the first place. But I hear you say, like, but Ernst, it's going to take years to develop a drug that will specifically upregulate Clothol. Can't we just kill them? Yes, that would be better, wouldn't it? Just get rid of the zombies. And you can do that with compounds that are called Xenolytics, so killers of senescent cells. And we are interested in those because they are really good drugs. So here's what I show you here is, this is again the amount of senescence, and it goes up over time. And these are these compounds, navitoclax, ABT7, dasatinib, and quercetin, very high in grapes, broccoli, and kale. Why is it always kale? I don't know. But they prevent the induction of senescence in brain organoids. So you can see here also that they are zombies because it goes up exponentially in time, just as you expect. I told you that senescent cells induce senescent cells in neighboring cells. So, okay, so that told us that brain organoid senesce and Xenolytics work. Okay, that's cool. There's therapeutics. They can slow the amount of brain aging, and in the next coming years, I'm sure these come on the market. And there are clinical trials ongoing now with D+Q to see if it inhibits Alzheimer's disease, and I think it's going to work. But now I'm going to tell you something about the virus we all love. And we have all grown to love, and the problem with it. And you know which virus that is. COVID. So you'll be sad to know that if you infect brain organoids with different strains of COVID, it induces senescence in brain cells. That's not good. And especially Delta is a particularly bad strain. We haven't tested any of the latest ones yet. But there are two possibilities here, right? So one is that the virus itself induces senescence, but we also know that COVID is more severely affecting older people that have more senescent cells already. So it's also possible that they preferentially infect senescent cells, and that's the reason why we see this. So we started to look into this. So what we did, we took brain organoids, exposed them to COVID, and then treated them with Xenolytics, these compounds that kill zombie cells, remember, and see which one worked. And they actually worked fantastically, especially D+Q, quercetin and dasatinib. If you look at the data, you can see here, this is P21, senescence marker. Basically, when you add D+Q, you basically come back to wild-type levels of cell that were not, brain organoids that were not exposed to virus. So not only that, it also inhibits the amount of virus that we have in the brain organoids. So these senescent cells are reservoirs for COVID-19 virus. So getting rid of them with Xenolytics, good idea. Just about to be published in one of the Nature journals. Start buying shares in D+Q. And this is where we use mice to see that we can do this also in a real-life system, because these were just brain organoids. Here we look at the survival of mice. Mice die rather rapidly when you give them a virus, this virus at high concentrations. Long story short, D plus Q and Xenolytics, they allow better survival, their clinical scores, their health scores go up, and the virus is also being eliminated. So it works in mice. So we need clinical trials in humans. Let me see the time. How long have I been going?
About 35.
Perfect. Perfect. So we also started to wonder whether we should be a little bit more sophisticated about our brain organoids. So at the moment, our brain organoids are a little bit of brain tissue, beautifully patterned and layered and thinking and doing the right thing, but that's not the only cells that are in our brain. We also have vasculature, we're working on that, but we also have cells that separate our brain from the spinal cord fluid. You might not be aware, but per day, 2 liters of spinal cord fluid is being produced by these layer cells called Choroplexus, and it basically washes your brain and washes out all the baddies and takes up nutrients and proteins that stimulate brain growth and health. So your Choroplexus is really, really important. They are sitting in the ventricles, these bits of fluid that are in the middle of your brain, and the Choroplexus sits right in between. Super active cells. So we thought, well, we should probably make brain organoids that have this layer. And here in red, you can see these Choroplexus cells. In white are the neurons. And when we go through it, you see that the neurons are on the inside. We show that they are still making the right patterning. And the Choroplexus makes cerebrospinal fluid and protects these brain organoids. Now, why am I telling you all this? Well, because the Choroplexus is really important for the SARS-CoV-2 virus to get into the brain. And the reason for that is that the Choroplexus is chock-a-block with the ACE2 receptor. You must have heard about the ACE2 receptor now, where all these antibody therapies for COVID are being directed to. That's the receptor where the virus binds to. Well, the Choroplexus, this layer of cells that separate your brain from the cerebrospinal fluid, is chock-a-block with it. And the funny thing is that people with Down syndrome have an extra copy of another gene called TMPRSS2 that is also involved in virus uptake, and they have more of that because on chromosome 21. People with Down syndrome have an extra chromosome 21. So we wondered, well, what had happened with Down syndrome brain organoids? Because people with Down syndrome have a 10 times higher chance of dying and getting hospitalized than the people that don't have an extra chromosome 21. So we did that, and we made Choroplexus ones, and basically what we saw is that the number of SARS-CoV-2 in Down syndrome was much higher, and that you needed the Choroplexus, this outer layer, to see that difference. And we also see that neurons are starting to fuse together. So these Choroplexus Down syndrome organoids can also tell us something about how viruses enter the brain and how you might be able to prevent that. It's disconcerting, though. And we also started to do similar tricks with other viruses that affect brain development. This is Zika virus. You might notice this is especially very prevalent in Brazil, and you end up with kids like that. Very small brain. Well, you don't have to be a rocket scientist to see what happens after you infect with Zika. You get a small brain. These are brain organoids. You can see that. And in this case, we worked with virologists at UQ, Alexander Kromick, who made a virus that didn't do that, but still elicited an immune response. So we are now together with them seeing if this can be a vaccine, because obviously this is a virus that behaves just like the wild type, but doesn't cause brain abnormalities, but still a good immune response. So this is one of these unexpected side effects of brain organoid research that you can like, oh, you can use it for that too. And yes, we are looking at Japanese encephalitis, at Lissa virus, at chikungunya virus, all the viruses that are in Queensland that are coming south. They're coming, boys and girls. And we're hoping to be a little bit anticipating and making model systems that we can test therapeutics before it becomes a problem. Okay, so COVID infects senescent cells and drives senescence in your brain. Stay away from this virus, wear masks, and get vaccinated. Xenolytics are a potential treatment for long COVID, and they are a beautiful model system to look at impact of viruses that affect the brain. Okay, so now let's start talking about some more crazy stuff. This is what we are doing. I showed you this, that you can put a brain cell on and start to look at activity. But we've also made heart cells that beat with the same frequency as your heart, and this is a calcium dye. And we also made kidney organoids that have the same plumbing and the same cell types as your kidneys. I told you they make all the cell type of the human body. So the next step now is to start to connect them in the dish. So we have little channels, microfluidic channels, that connect all these organs and with an artificial bloodstream. And now we might be able to simulate how microbiota in the gut affect the brain. And we know they do. Or we can start to look at the effect of drugs that first go through a liver and then affect the kidney. I mean, that's pretty cool, right? These are human cells, these are your cells. I think this is what we need in the future in our hospitals. I can see a future where you're born, you donate your cord blood, we make these iPS cells from them at the start, really cheaply. We have them sitting there. They can be your repair kit for cellular therapy to replace cells after you had a motorbike accident. But you can also see them as personalized screening tools for when you get a cancer or when you have a particular genetic disease that you didn't know you had, but which you only found out when you were 21. That's what I see as the future. So, no ethics at all here, right? I mean, they are just, of course there are ethics with this. In a time when we used embryonic stem cells, this was a real question, right? But now, I told you, is that these cells can be made from a blood cell, but they are functionally equivalent to an embryonic stem cell. For those who don't know what an embryonic stem cell is, these are these same pluripotent stem cells that make a whole embryo, make all the cell types of the body, but come from surplus IVF embryos. So we don't know whether they would ever have gotten the disease, but that's where they came from. And you can remember they are banned in the United States, they're banned in Germany, Australia. We are enlightened, we are allowed to work with them. But since we can reprogram to induce pluripotent stem cells, which are exactly the same as embryonic stem cells, we hardly use them anymore because the personalized stem cells are much more powerful. And as I told you, the problem with these organoids, they're all about pea size or lentil size. And why is that? Well, you can sense why, right? Because they just can't get enough oxygen, they can't get enough food. The diffusion limit is the barrier. So everybody is trying to engineer organoids with vasculature. And like we can make vascular cells, the problem is to make them perfusable with blood. That's a real engineering challenge, and we're not there yet. But I can tell you, as soon as we can do that, these organoids will become bigger. They will become close to life size. So that's going to be an interesting time, right? Maybe you will have an ability to make replacement organs somewhere in the future. Now, we're already playing these kind of games, right? This is from my laboratory, where we take a human brain organoid and we scoop a little bit of a brain out of a mouse that doesn't have an immune system, so won't reject the human graft. We pop in a human brain organoid, and because it doesn't have an immune system, the mouse goes like, brain, yeah, I know that. And it will just integrate into the mouse brain, it gets vascularized, it gets a blood supply, and it functions just like a human brain in a mouse background. No ethic problem at all, right? It's kind of important, right? Because if we find drugs that work on brain organoids, we would like to test this in a very small amount of mice and say, like, okay, even when this brain organoid lives in a real, wet brain environment, the drug still works, the drug still gets to the right spot. So it looks a bit weird, and these mice are not smarter. They don't read the paper or anything like that. But it does blur the line between, well, at what percentage of brain organoid is this still a mouse, and what percentage is it becomes human, right? There's a limit there, and we, together, need to talk about where those limits are. Because what I haven't told you yet is this. I showed you that they develop this rhythmic activity What you don't know, well, maybe some of you do, is that if your brain starts to show coherent activity in the 100 hertz range, so 100 times per second up and down, that's called gamma waves. And when you go under anesthesia, you lose the gamma waves. You keep the other ones mainly, but you lose the gamma waves, and you don't remember or feel a thing, right? Well, you can see what's going to happen here, right? These brain organoids start to show, after about 7 months, they start to show gamma wave activity. No ethical problem at all. But it's kind of interesting, isn't it, that a clump of human brain cells that came from a blood cell that were reprogrammed into a cell that was a day 5 embryo from somebody who could have well deceased, is now able to make brain organoids that show autogenic activity. I think they're dreaming because they don't have any connection with any senses, no hearing or visual input. But they by themselves, after 7 months, just when embryos in vivo start to show gamma wave activity, that clock ticks in real time, ladies and gentlemen. These brain organoids that start to show gamma wave activity, and that's akin to consciousness. I mean, I'm intrigued, right? I mean, consciousness is something that we don't even know what it is, but we can see signs of it, and it emerges by itself just because it's a complex, multi-component, interacting system of human neurons. Other species do this as well, okay? So maybe we're not the only one that have consciousness, if gamma wave is the only readout. But the good thing is, what we're doing at the moment, we're starting to screen for better anesthetics, because anesthetics are not necessarily good for you. So we're trying to find better anesthetics. But we're also trying to find out which cell types, or which combination of cell types are underlying this gamma wave activity, because I can't resist. Now, I do think about the ethics of this as well, and this is what we published in the American Journal of Bioethics. And this is a brain organoid, not made by us, and this is not just a trick. These are indeed proto-eyes. These are optic cups that have been engineered to be on a brain organoid. And what they have shown, not us, others, if you shine light on these optic cups, the visual cortex on the back of this brain organoid starts to light up. So this one can see, right? So this one does have sensory input. So we can connect this, we can make combinoids, we can fuse this to a hypothalamic organoid, to other organoids. We are making spinal cord organoids that are matching this to see how that connection works. You can see where this is going, right? At some stage, we're going to have to say, like, maybe they have rights. And especially this freaked me out. And we're doing this ourselves. So this is, in an effort to understand how our brains learn, this is real. We have brain organoids in our lab that we're putting on our multi-electrode arrays, and they play Pong against each other. And they learn this in a couple of minutes. AI takes like 5. Sucks AI. But our brains are pattern recognition machines. That's how we evolved, to see like a bit of a tiger tail there, an ear over there. Tiger, I know. That's how we evolved. And if we give these brain organoids, even though they're really simple, very simple commands, go like, here's a bat, there's a ball, bounce. It goes like, yeah, give me something a little bit more difficult, right? But we're starting to use this to interrogate, like, so what happens when we put them slowly under anesthesia? How do these brain organoids learn? Because that's fundamental to what makes us human. And that's what I like about this, right? Curing diseases, fantastic. Drug screens, awesome. Better drug pipelines, fantastic. But this is also kind of cool. I want to find out. Okay, do I have 5 more minutes? Perfect. Okay, I promised you I was going to talk about the elephant in the room and why we did this, okay? So this came about because we are stem cell biologists. when a synthetic meat company from Sydney called Vow contacted us and said, like, can you engineer these sheep cells to grow forever, just like you do with your human cells? And we go like, yeah, no problem. So we engineered them with an enzyme called telomerase that allows these cells to grow forever. And they were like, okay, that's cool. Now, can you do this for us? Can you make sure that these will make mammoth myoglobin? I go like, should be doable. So I am going to ask these guys to run the video. This was a crazy collaboration with the third biggest advertising company in the world and Vow. As I told you, they are a leader in synthetic meat.
The Mammoth Meatball brings back to life an extinct species to start a global discussion about a more sustainable food future. Because the current meat industry is destroying the climate, and in its wake, 1 million species. Could we turn the mammoth, extinct because of climate change, into a symbol of hope? Together with bioengineers, we identified a gene called myoglobin and used public data to identify the DNA sequence in mammoths. We filled in any gaps with African elephant DNA. We then inserted the full genome in carrier cells and grew 20 billion of them. And voila, 4,000 years after it went extinct, the mammoth makes a return. As a meatball, the unveiling in Amsterdam sparked a global discussion.
Next on the menu, mammoth meat.
I've been dying to know what a woolly mammoth tastes.
Could you eat a mammoth meatball and remain vegan?
Meatball from Mammoth. Made with actual mammoth meat.
The world was buzzing. The mammoth meatball was discussed on leading news channels, at dinner tables, and in classrooms everywhere.
Okay, that's enough.
I wonder how woolly mammoth tastes.
It is a giant meatball. Come on, it's it's a mammoth meatball. But I like the idea behind it, right? This was an advertising gimmick, right? Because we were not immediately going to eat this, and I know somebody's going to ask. No, we did not taste, but I did smell when they made it. Oh my God! I can see why they hunted it to extinction. It is umami, meaty, gamey. I would eat it. But we didn't want to do that. But it does show you that stem cell science, because these are myoblasts, these are muscle stem cells from the sheep, that we engineered to have this myoglobin protein, which gives the meat its taste. And it suggests that we can do this for other species as well. And I have been asked this after the whole craziness of the mammoth meatball. Like, can you make T-Rex burgers? If we find some DNA of a T-Rex, then we can fill it up with some salamander, and then maybe, yes. I'm not sure that we're going to be that tasty. But it is another unexpected application of stem cell research, right? And I think that's why I was on board with this company. We do need to start thinking about the environment and make meat in a more sustainable and also ethical way, because there's no mammoth were hurt in the process of making these meatballs, clearly. Anyway, so it does open up a whole Pandora box. So I think I'm going to leave it there, because I want to give plenty of time for questions, but I do want to acknowledge all these amazing people. This is my lab. Fantastic, multicultural, multi-gender, multi-skilled team, amazing people to work with, fantastic collaborators at UQ. These are the virologists, these are the spinal cord guys, this is the neurologist that looks after the epileptic patients, this is our mission, these are our funders, that's our lab, that's our building. Come and visit. I love to talk to people that are interested in this, and I hope I've given you a flavor of where the possibilities are, where the caveats are, and where the cool things are going to happen. So I'm going to leave the robot to work, because it's working right now, while we're asking questions. Cheers.
Wow. What a tour de force. We've got science, investing, ethics, zombie defense tactics. Meatballs, that's right, dinner tips, and serving tonight at our refreshments afterwards. No. Okay, so now's your chance to pull out your question app, ask questions. The QR code is up there if anybody missed it at the start. And we're going to get into a few questions here. I'm just going to ask the first one while I'm looking at this. How much of the woolly mammoth DNA did you get, and how much needed to be filled in? Yeah, we've all seen Jurassic Park, and we know,
It was about 85%, if I remember, 85% complete. So the vast majority was mammoth, because they're finding mammoth in the tundra, right? These frozen mammoths, so they're sequencing them. So we got a pretty good DNA source.
And why didn't you eat it? Is there science behind that?
Because the company is very risk averse, obviously, because they have just launched their what was it? It was quail and scallop mixture. So putting quail and scallop proteins in the same cell. So they went like, well, that we've tested. That's one. And secondly, because our immune system hasn't seen this protein for like 16,000 years, it's possible that we have some weird allergic reaction to mammoth, right? So that's why we said, like, look, let's first test it, and then we will eat it. But we've had a lot of requests go like, I'll take the risk, give it to me now.
I'm pretty sure that's going to be the next Marvel superhero, Mammoth Man who eats a radioactive meatball. All right, we've got a lot of questions here. First question is from Katie. How do cells know how to construct a 3D brain? Do we understand how disease interferes with that? I think there's a couple of other related questions there.
How do cells know? It's a really good question, Katie. So they know this by virtue of their DNA, right? The DNA sequence, and obviously the DNA in your skin cells and in your brain cells and in your liver cells is all the same. So it's not just DNA, but it's the way that the DNA is accessible to be read, and that's what's called epigenetics. So there's a program in each of these stem cells that says, okay, so cupboards 1 to 4 stay closed, cupboards 3 to 5 open up, but just books 12, 18, and 15, and that's the recipe for a brain cell. So that's how cells know. It's the epigenetic regulation of the stem cells, and over time that changes, that tells the cells how to behave. At the same time, they are seeing their neighbors. This is why this works best in 3 dimensions. They are seeing their neighbors, and they go like, okay, so I'm a cortical neuron, you are a layer 2 neuron, we stay. And then the cells, a brain is built inside out. I didn't tell you that. Well, you saw that from the picture, right? The stem cells are in the middle, and the cells migrate through the layer and then lay down a layer. And then the next one migrates through and lays down the second layer. So every time when those cells migrate through and that epigenetic program is running and it sees that it's got the right neighbor on the bottom but nobody on top, it goes like, okay, got to become layer 3 clearly. And then it does. So that's how nature, creates complexity with very simple instructions.
Fantastic. All right, we're jumping around a little bit here. Gareth asks, can you take my cells and create stem cells that will replace disintegrated cartilage in my hips?
Yep.
And I might just add, it's probably a little tangent here that Sith asks, will treatment using stem cells have risk to cause cancer or tumour? Are there risks? Is it safe?
Yep. So 2 really good questions. So one, yes. I mean, that this is already being done with our footy players, right? so there you can harvest bone marrow cells or fat cells and turn them into what's called mesenchymal stem cells, which are cells that make bone, cartilage, or fat. So if you give them the right cues, they will make cartilage. The problem at the moment is to tell them to make the right cartilage, because not all cartilage is created equal, and usually you need a scaffold to tell them where to go and where to stay. But having said that, mesenchymal stem cells are also fantastic anti-inflammatory cells, so this is one of the ways that they may actually benefit people when they get injected. So yes, you can repair cartilage with mesenchymal stem cells, and you can make mesenchymal stem cells from these reprogrammed induced pluripotent stem cells. I forgot for the second question.
Safety and risks.
Yes. So this is a problem, something to keep a real close eye on, right? Because cancer is a stem cell disease. I mean, you may not be aware, but this is with the nucleus of cancer. These are stem cells that are just keep dividing but don't become the right cell type anymore. So yes, you do have to be super careful when you make billions and billions of cells that you don't have cells that have mutations that will make it cancer. But there are ways to engineer it that it becomes safe. For example, you can engineer suicide switches in cells that as soon as they become a cancer cell and they start dividing when they're not supposed to, dead. So there are ways around it, but it is an issue.
Now, we are a little over time. Is everyone up for just a few more minutes of questions? Because I've got like 500 here. If you want your take that and a packed lunch, get up a chair because you're going to be here a while.
This is fun.
Great. All right, we've got a bunch of people, Din and others, who've asked, what is the process by which you convince stem cells to go down a particular path to differentiate into certain types?
Yeah, you said we had a good audience, and we do. That's a really good question. So these are the same cues that cells use in a real-life developing embryo. These are usually proteins that are being excreted by cells and diffuse to target cells, then bind to a receptor and initiate a signaling cascade. So basically it's the right combination of proteins, but more and more now it's small molecules like chemicals that mimic the action of those proteins. Otherwise it just becomes too expensive. So it's basically activating signaling pathways in the cells at the right time. So this is what we do. We add a little bit of sniff of this combo here for 2 days, remove it, add another combo for 2 or 3 days, remove it, and then usually once the system is on its way, we can let it go.
Slight follow-up. Are the stem cells developed, the cells developed from pluripotent stem cells, are they the same as those that occur naturally in the body?
Yes.
And functionally and all the same?
Yes. Yes, absolutely. And the beauty is that if I would make them from you, I can inject them into you because your immune system goes like, mine.
Amazing.
Doesn't reject it.
I'm excited. All right, we're on the last few. Last few. We're talking the last 3 of 100 here questions. So a couple of questions around how do you dispose of your organoids after the fact? Somebody asked if you cut them in 2, do they grow like worms? But let's.
Well, the funny thing is, if you cut them in 2 early enough, they will repair themselves and make 2 twin organoids, and that's actually something that is also really interesting concept. We know this from the heart, and we're investigating this in the brain now, that if you injure an embryo really early during development, it will heal itself scarless. As soon as an embryo is born, and this has been well established in the heart, as soon as it's born and it sees oxygen and it's out there, then the same injury doesn't repair and you get a scar. So something happens, there's a switch somewhere just before birth where we lose this ability to regenerate. Now, axolotls and salamanders, you cut off an arm, it regrows, right? So we are trying to look at that and hack that, investigate it in brain organoids, making injuries over time until they're 9 months. And then what we think is that they lose this ability and then find out what's happening and then hack that so that we can impose it on adult stem cells and repair brain damage. So, yep.
Amazing. And I am pretty sure that was the plot of the 4th Spider-Man movie, but yep, just saying, science fiction writers, you're having a great night here. Okay, Jacob asks, second last question, how far are we from bringing back extinct species?
I think we're very far away. So I've been asked this question with some of the Russian oligarchs when they were still in the good books, wanted to do this. So you could do what's called something that's called somatic cell nuclear transfer, where you take a nucleus, what they did with Dolly. You could take a nucleus from a mammoth that is well frozen, take an elephant egg and replace the nucleus of that egg with the nucleus of that frozen mammoth. Do that probably several thousands of times, and then maybe one will develop into an embryo. Then the problem is, now you have to make 100 elephants pseudo-pregnant, implant those 100 mammoth embryos that you may have made, and then hope that it will come to term and not become a monster. It's not ethical, right? So that's the only species that I know of where we have almost a full genome. We've been looking for a genome of the dodo. That would be nice. But having said that, if we have it, it's now very easy to synthesize genes artificially. So we don't have to clone it anymore. You can now go to a company and say, like, give me this sequence of bases, and they'll make it for you. So in principle, if you have the whole genome, you could assemble it, but then you still need all the regulatory sequences. Long story short, I think it's just going to be very difficult. And do we really need it? But let's look after the animals that we actually have.
Great message, which perhaps leads to, there are a whole bunch of messages here about wanting to know how to work with you, whether you have internships, where can they go buy your coffee, and one that looks pretty much like a wedding proposal.
Sorry, take it.
I'm not kidding. So, yep, sorry, you got your answer to that one. So, what's the next steps for all those young stem cell first years who want to do this, as their future? What's your advice?
Yeah, contact me. If you got good grades and you got good ideas and you want to work 6 days a week, and you're not afraid of being challenged, then contact me for sure. I mean, we have lots of opportunities for students, and we welcome new thinkers, new ideas from all corners of the world, from all corners of the spectrum. So yes, bring it on. Come.
Amazing. Well, look, I think we will absolutely want to have you back in. Please, huge round of applause for Professor Ernst Wolvetang from UQ. It's amazing.
Oh, thank you.